Hi everyone. Could we please scan the attendance QR code just to verify that you were here? We're going to start in around 2 minutes.
We're fixing it, do not worry.
We'll just start. Until people do. Sorry guys, we heard the form doesn't work, so we're going to try and fix it, and after the presentation ends, we'll have everyone scan it. But for now, please welcome Aiden to present Products of Economic Development and Agricultural Pollution.
I'm presenting on products of economic development and agricultural pollution, rare earth element inhibition of cell growth. And this paper is by Anna Kotelnikova et al. And it was published October 4th, 2018. The title of this paper is Toxicity Assay of Lanthanum and Cerium in Solutions in Soil. So rare earth elements are the elements on the bottom 2 rows of the periodic table, and they can be found in phone screens and in a lot of different industries such as chemical processing and catalytic converters. But overall, despite the name, they're not that rare because they're abundant in the Earth's crust. And so production of rare earth elements over time has dramatically increased, as you can see by the huge increase even from just 2000 to 2010. And the problem with this is that rare earth elements accumulate in the environment. So even if the rare earth elements are contaminating soil or just simply algae, as it goes up in the ecosystem, it accumulates and comes in higher concentrations and toxicity to humans. And so what's important is the food cycle, because let's say the rare earth elements inhibit the acorns, then the squirrel will no longer have food to sustain life, and then if the squirrel can't sustain life, then the fox can't sustain life as a result. And so it's a chain of events, and so if you inhibit one thing and take one thing out of the ecosystem, it affects everything in the ecosystem. Another important thing to know before I get into the figures is mitosis, and the most important thing is just that it's the cell division process. And then
There's a second leg in the box show.
push it on. And then some other things to note are different abnormalities in the chromosomes and in the cells that have rare earth elements. So the first one is chromosome stickiness, which is when chromosomes get stuck together, and that results in the chromosomes breaking, which can lead to aneuploidy. And then chromosome fragments, which is when chromosomes get broken because of stickiness or some other abnormalities, but basically you have just fragmented chromosomes that will produce irregular proteins as well. And then lagging chromosomes, which is when chromosomes get stuck on this spindle in the center, as you can see with this chromosome, and that can also lead to chromosome fragments or aneuploidies. And then the anaphase bridge is when DNA thread forms and results in aneuploidy or polyploidy, and you can see the bridge between the chromosomes here. And then the last one is colchicine mitosis, which is a drug-induced effect where the cells get stuck in metaphase, and you can see right here all the cells are in metaphase as opposed to being in the varying stages of mitosis. So in this paper, what they did is they studied the effects of rare earth elements on onion cells, and they specifically used onion cells because they have 16 large chromosomes, rapidly growing bulbs, and large cells, which makes it easy to observe the effects of the rare earth elements on the growth of the onions. So they observed mitosis, the normal mitosis versus mitotic abnormalities through computer platforms and through microscopes. And then that eventually led to them quantifying the data with 3 different quantifications. And the first one is the root length, which basically shows how much the onion root has grown in different concentrations of rare earth elements. And then the second one is mitotic index, and what they did is they quantified the number of cells in mitosis and then divided it by the total number of cells and multiplied it by 100 to get the percentage of cells that are in mitosis out of all the cells. And then lastly, they studied the frequency of aberrant cells, which is basically how frequently they see chromosomal abnormalities in abnormal cells. And another important thing to note is that although this figure only shows soil, it's also important to note that another thing that they tested was in solutions, and for each solution and concentration in both solution and soil, they tested 5 different concentrations. In figure 1, it shows the growth of bulbs in solution with lanthanum and cerium, and an important thing to note is that the x-axes are different values. So for lanthanum, it ranges from 0 to 200 milligrams per liter, whereas for cerium, it ranges from 0 to 50 milligrams per liter. And that's because cerium tends to be overall more potent and more toxic compared to lanthanum. So they wanted to make the y-axes relatively similar so that they can observe the effects of lanthanum and cerium. And so for root length, and this is in solution, so for root length, you can see that in lanthanum, overall, the general trend is that the root length is decreasing, showing that lanthanum and cerium have inhibited growth of the roots of the onions. And that goes the same for cerium. And figure 1C and D show the concentrations effects on mitosis. And you can see overall the general trend is that the percent of cells in mitosis decreases in both lanthanum and cerium. And figure 1E and F shows the concentrations effects on the frequency of aberrant cells, so how many chromosomal abnormalities it may have in different abnormalities in the cells. And you can see that from 50 to 100 milligrams per liter, it starts to decrease, and that's because the error bars are pretty significant and the data really can't yield that many results, but you can see by the asterisk above 50 that that result is significant because it's pretty different from the control, which is 0 micrograms per liter of lanthanum. And overall, the general trend, the percentage of cells, the aberrant cells, is increasing. And the same goes for cerium. Figure 2 shows the growth of bulbs in soil with the samples in concentrations of lanthanum and cerium as well, except an important thing to note is that the x-axes are the same for this figure, and that's because the researchers found that in soil, the rare earth elements seem to be less bioavailable as opposed to the solution. And that's probably because there are varying nutrients and varying nutrients that you can find in the soil as opposed to just having distilled water. And so in figure 2a, you can see that it decreases and then slightly increases from 10 to 20 milligrams per kilogram. And that can be contributed to the fact that lanthanum and as well as cerium are naturally abundant in the Earth's crust and are necessary for plant growth and plant life. And you can also see that the general trend for lanthanum seems to be pretty constant, showing that in soil, lanthanum has less of an effect on the root length of onions and doesn't inhibit growth quite as much as it does in solution. Whereas in cerium, it is a pretty negative slope on the trend line, showing that overall cerium in the higher concentrations, it inhibits growth of the onion cells and the root lengths will be shorter. And this figure shows lanthanum and cerium concentrations effects on mitosis. And you can see that there's another slight increase in the percent of cells in mitosis, and that can be attributed to two things, either human error due to the error bars, or it can be attributed to the fact that lanthanum is naturally abundant and necessary for plant growth. And the same goes for cerium. So in both lanthanum and cerium, the same slight increase is observed from 50 to 100 milligrams per kilogram. However, the overall trend is still slightly negative, showing that the rare earth elements inhibit mitosis and the number of cells in mitosis decreases. And in figure 2E and F, it shows the concentrations effects on the frequency of aberrant cells. And you can see that after a certain point, which is 50 milligrams per kilogram, the effects of lanthanum becomes pretty constant, and that's because there's a certain threshold as to how much toxicity these cells can experience, and that's pretty much the maximum. However, the overall trend, it's still slightly increasing, the amount of aberrant cells. And then for cerium, overall it's just pretty constant, showing that cerium does not really increase the amount of aberrant cells. And in figure 3, they basically help visualize the abnormalities of mitosis in onion cells. So what they observed in figure 3A is chromosome stickiness, as you can see by the chromosomes being relatively clumped and entangled together in the blue box. And then in figure 3B, you can see disturbed anaphase, and then figure 3C, you can see disturbed anaphase with a vagrant chromosome, which is the chromosome that's stuck in the center. And in figure 3D, you can see the lagging chromosome in anaphase, which is in the cell in the blue box, and it's this little chromosome that's stuck to the pole of the cell. And then in figure 3E, you can see disturbed metaphase again, and then in figure 3F, you can also see disturbed metaphase. And so the limitations of this study are that the error bars are extremely large, and also they only tested 2 types of rare earth elements. However, lanthanum and cerium are the most abundant in the Earth's crust. And in addition, they added Cl3 to form ionic bonds with the lanthanum and cerium in order to increase the bioavailability and absorption of lanthanum and cerium. And then, again, they have some unusual trend lines for some data that should theoretically show that lanthanum and cerium do decrease plant growth. However, some of them show just constant lines, and that may be due to human error. And a solution for the accumulation of rare earth elements in the soil is bioremediation, which is some ways that you can do this are by adding chemicals to soil, or another proposed method of decreasing the concentration of lanthanum and cerium that's found in soil due to pollution from the big tech industry is by adding composted kiwi fruit branch, which is pretty interesting because it can neutralize the toxicity of lanthanum and cerium. So overall, high concentrations of rare earth elements lead to cytotoxicity and overall anti-proliferative activity, as well increased frequency of aberrant cells. And so things that we need to do are moderate high concentrations of lanthanum and cerium that we find from pollution from the tech industry. And we need to be proactive, not just reactive, by doing things that will prevent the accumulation of rare earth elements in soil. And as well, this is a pretty specified study, so it has a very specific impact, but it's very relevant because of the growing tech industry. And the studies show that naturally you can find up to 100 milligrams per kilogram of lanthanum and cerium in soil, maximum, but then with humans you can get up to 1,000, which is very significant. So acknowledgements are my journal club staff that I worked with, which is Julia, Sarah, and Zoe, as well as Mr. Maxwell and all the teachers giving extra credit. And thank you to Bodega for coming to this presentation. Are there any questions? Eric?
Do we know how, exactly on a molecular level rare earth metals actually mess up these mitosis?
I don't personally know, but I assume that it, I don't know the underlying cause, but you know that from the article they talk about having chromosome stickiness and chromosome breakage, so something to do with lanthanum and cerium being toxic to, it has oxidative effects. Angelina?
What do you think the next steps for these researchers?
I think that they should study perhaps a greater sample size because the small sample size led to pretty large error bars. And I also think that they can study different types of rare earth elements as well as different types of plants other than onions because there are some plants that show to absorb rare earth elements and help with soil remediation. And so if they tested those, that would be very useful. Ryder.
Does the lanthanum and the cerium specifically affect the mitosis abnormalities?
So lanthanum and cerium have oxidative effects on the onions, and that leads to having abnormal mitosis, and then the chromosomes break, and you have different chromosomal abnormalities, and that leads to weird protein production, which inhibits the growth. Thank you.